All right, this is part 4 of chapter 11, microbial genetics. And this one will talk about a few things. We'll talk about plasmids and gene transfer and operons. So what's a plasmid? Ooh, sorry, guys. A plasmid is an extra chromosomal. So it's not part of the bacterial chromosome. It is outside of it, hanging out inside the bacterial cell. They are little circular pieces of DNA, and these are always going to be sort of beneficial for the bacteria. They give the bacteria extra genes that make them either capable of gene transfer or they can make them antibiotic resistant. We'll talk about antibiotic resistance later, but they're called R plasmids, R for resistance. And then there's one in particular we'll talk about called an F plasmid. An F plasmid is for fertility. What does that mean? So it turns out that there are 3 ways that bacteria are able to sort of have a way to have different genetics, like mix up their genes, have like basically it's called genetic recombination. They get genes that are not originally part of their cells. It's from another cell or outside. So let's go through the 3. Letter A is transformation. We talked, no, we didn't talk, we talked about this one. So transformation is simply that some bacterial cells, this guy right here, that's a plasmid, okay? That's a plasmid. It's a little piece of circular DNA, plasmid. Oh, sorry. You guys know what I'm doing, plasmid. And it's just hanging out in the environment. So maybe this came from some bacteria over here that died. It died and it had that plasmid, and then it basically just, the plasmid came out as it was dying, and now we have this little plasmid hanging out. Some bacteria are just able to absorb it. They can just take that guy in, and then they can incorporate it into its own genome. They can also just keep it as a plasmid, but they just snatched it up from the environment. So that's called transformation. They can just pick up DNA directly from the environment. Transduction is where a virus infects a cell. We talked about this in chapter 6. So if you need to review transduction, review the chapter 6 area on viruses. But the viral DNA can come in, and the viral DNA can incorporate into the bacterial genome. And now the bacteria has this extra information here that can, it does code for virus, but also, if you remember, there was like that specialized transduction where if this virus had infected another bacterial cell and it contained a small little bit of that first bacterial genome, it can also transfer that little bit of that bacterial genome into the second bacteria that it's infecting now. So it can get bacterial genome. Some viruses actually make toxins that are encoded for on their genome, and they can basically just give that bacteria the information to make a toxin, and now that bacteria can make this like special toxin. So this is another way that we can change the information that the bacteria has inside the genome. And then there's a third way, and this is where the F plasmid comes in. Sometimes bacteria can just literally make this little bridge between the 2 cells. They come really close together, they make a bridge, and then within this bridge, the plasmid will copy itself, right? Replicate itself, and just donate the plasmid to the next cell. Okay, kind of crazy, but that's called conjugation. So let's learn some terms that go with conjugation. So the bacteria that happen to have an F plasmid, F is for fertility, is called an F plus donor. This guy is able to make a pilus. This is also called a sex pilus. But basically it's the ability to make that bridge. Not everything, this bridge right here, right? So not all bacteria can make that bridge. This is very much the property of that F plasmid. And when you make that bridge, what happens is the F plasmid could copy itself, put a copy into the second cell. The second cell without the F plasmid is called an F minus, right? It doesn't have the F plasmid. But after conjugation, we have 2 F positive cells. We have this cell, this cell copied the plasmid, right? So it didn't give it its plasmid. It copied it. It gave a copy to this F minus cell. So after conjugation, we have two F plus cells. Now both of these guys can go on and form a P-less and send that F plasmid to another cell. What is clinically significant here is that not only does the F plasmid go over, but this bacteria that's F plus can also send over other plasmids that it may have, right? So if it had like an R plasmid that coded for resistance to penicillin, right? This bacteria with an F plasmid can actually also donate that resistance plasmid to the recipient. It can also take some of its genome and give some of its genome to the other cell. So it's a way that bacterial cells can actually have this genetic recombination, because bacteria, you know, they divide by binary fission. It's asexual. They're making clones 100% of the time. But to create some variation in the cells, in the population, there is this evolution of this ability to share information between cells. And the F plasmid is key to that conjugation. Now, there is a special kind of circumstance that if the F plasmid actually inserted itself into the genome and just became part of the bacterial genome, then that cell is called an HFR. Male, let's just do quotes, right? Male. HFR stands for high frequency of recombination. That means that this cell now is really into conjugation and will form that sex P-less and donate not only the F plasmid, but basically pieces of the chromosome too, right? So don't worry about the example here, but if there's a lack, gene, that gets put into our recipient. And then now our recipient has the F plasmid and the extra genes. So so that's it. So the HFR cell is basically where occasionally that F plasmid is incorporated, and then it can lead to more recombination, more conjugation, and you can transfer the F plasmid along with other plasmids that way. So it's just like a turbocharged F plus cell. Okay, so here's another image of an HFR cell. This is showing you that not only does the plasmid go over, right, so the F plasmid is incorporated in this bacterial genome, and the HFR not only sends over the F portion of it, but also sends over, a lot of its genome. So this recipient cell is going to get a lot more information, and maybe, new bells and whistles. Maybe it can now, make a toxin that it couldn't be able to before. Now it makes some, something different. The phenotype has changed because of that chromosome coming over. And so that's basically it. Don't worry about this picture. We're not going to really talk about that picture. All right. And then the last subject in this is an operon. So an operon is only in bacteria, okay, only in prokaryotes. It is because the bacterial genome is so much smaller than ours. Remember when we talked about like there's a lot of non-coding DNA in eukaryotic cells, and there's like genes that are like far apart from each other, and they have a promoter region and they have a terminator region. For bacteria, they want to save space. So what they do is all the genes that kind of work together that do the same job are going to be grouped together right next to each other, like back to back to back to back. So all of these genes are controlled by one promoter. There's an operator. We'll talk about that. And then, so it looks slightly similar to our genes that we talked about before, right? There's a promoter region, then there's a termination sequence. So when RNA polymerase comes and, attaches to the promoter region, what happens in bacteria is that the RNA polymerase will come bind here. It will move down the line, and it makes gene 1, it makes gene 2, it makes gene 3, it makes gene 4, right? So we're making the mRNA for all 4 of these genes at once. And then we get protein A, protein B, protein C, and protein D all together controlled by this like one promoter region. So we'll look at a specific example, but again, an operon. So if I want to define an operon, what is an operon? An operon is several genes being controlled by one promoter region and one operator, okay? And these are all expressed together. You can't just express A. You're going to do A, B, C, and D because they all have a similar job to do. They all work together to do a certain thing inside the cell. So that's an operon, okay? The specific operon you guys will learn is called the LAC operon. LAC stands for lactose, okay? So we'll talk about specifically E. coli. E. coli likes to eat, primarily, it loves glucose. So number one yum food for bacteria, this E. coli, is glucose. If glucose ran out, right, and there's no more glucose around, it can go to food number two. Food number two of choice is lactose. So basically what happens in this operon is that the enzymes and the molecules that work to bring lactose in the cell, like these channel proteins, they are not going to be expressed most of the time, right? Because why would they? Because if there's glucose around, glucose is going to be used by the bacteria. The E. coli is going to be happy. There's no reason to express any of these genes that will help break down lactose. These genes in the LAC operon are specifically to break down to bring in more lactose and to break down lactose. So what happens is there is something called a repressor molecule. Okay, a repressor molecule is going to physically sit on this operator area and block the passage of RNA polymerase. So RNA polymerase can bind to the promoter, fine, but it's not going to be able to transcribe these genes into RNA because there's this physical repressor sitting there. Okay. So this is basically the off position. If you want to talk about like operons being on and off, this is the off position. Okay. So what this is called is the LAC operon is an inducible operon, meaning that it's going to be off most of the time because of the repressor sitting there, because lactose is its second choice, right? And only turn on, okay, only turn on if we ran out of glucose and now we have to switch to lactose. So in that case, we need to remove the repressor. But how does the bacteria know when to remove the repressor? So there's this little binding site on the repressor, and that is for a molecule called the inducer, and in this case, it's lactose. So lactose, as it trickles into the cell, will bind to the repressor. The repressor then changes shape at conformation and it falls off the operator area. When it falls off, there's nothing blocking RNA polymerase anymore, and it can go ahead and it can transcribe LAC-Z gene, LAC-YZ gene, and LAC-AZ gene. Okay, so these genes all have a job in metabolizing lactose. So one of them is going to be called beta-galactosidase. Beta, it's called it's written in your book, galactosidase. It's a long one, galactosidase. So it's an enzyme, and what the beta-galactosidase does is it breaks lactose down, okay? So if you remember, lactose is a disaccharide. It's two sugar groups hooked together. And in order for the E. coli to use the energy here, it has to break this bond between the lactose monomers, right? So this enzyme, that when it's made, will start to break down lactose. So you can think about that's really great. And then there's another there's another gene that codes for something called permease. Okay, permease is a channel protein. And permease will put up, I'll draw it up here. So if you imagine the bacterial cell, like this is E. coli, and there's all these lactose molecules in the environment, right? The lactose molecules, some of them at a slow rate will diffuse into the cell, but slowly, okay? What permease does is it's a channel protein. So when LAC-Y is being made, it'll come up to the surface, little like basically tunnels, right, that allow lactose to go through. So permease allows much more lactose to come into the cell than it would through diffusion. And it allows the cell to get all the lactose it can, and then the beta-galactosidase breaks it down, and then the cell can use it as energy. Okay, so that's the gist of the promoter. The lac operon, it is a, let's just say I'll put it down here. The lac operon is an inducible operon, meaning that, like when you induce labor, right? You're not in labor yet, you have to induce it. So this is off most of the time. So you have to induce it to come on, right? So that's why it's called an inducible operon. It's off. What is the inducer? What is the molecule that says, hey, let's have this operon turn on and express the genes? The inducer is lactose. Okay, the inducer is the thing that removes the repressor and allows the expression of the 3 genes that we have in a row. We won't talk about the last gene, but I just want you to know that one of them is for beta-galactosidase. Oh, I didn't finish the word, sidase. I just wrote side. Beta-galactosidase, the enzyme that breaks down lactose, and then permease is the channel protein that just allows more lactose to come in really fast. So let's look at this chart, right? So when E. coli is grown in the presence of 2 substrates, we have glucose and we have lactose in the medium. E. coli prefers glucose. So what happens is E. coli grows really, really fast. It uses all the glucose up in that medium. And then there's a point where it stops growing, because what happens is the glucose is gone. It ate all the glucose, and then there's this stop. There's this like time where the E. coli is not growing anymore. There's like plateau, right? It's not dividing. In this time, the bacteria has now needs to shift over to lactose metabolism. So in this time, the cell is going to be expressing the lac operon, because lactose is present. It's just present. It's present, right? So it'll be there. But since bacteria really want to use glucose first, it will not turn on the lac operon until all the glucose is gone. And this is actually pretty complicated. There's other mechanisms at work here, but we're not going to go into that detail. But this lag period is when, okay, we got to get the lac operon going. We got to make our beta-galactosidase. We got to make our permease. And then the bacteria starts to grow again, right? You can see this up, because now it's using lactose. And from just the shape of the growth curves, the E. coli grows much quicker. It has like a steeper upslope than with lactose. Lactose is like a little bit not as steep, so not as fast, but it still grows, right? So, so that is basically a visual of the preference of bacteria to use glucose first, lag time where we have to get the lac operon going, we have to make the permeases, we have to make the enzymes, and then we can start to use our lactose and grow again. All right, almost done. So for eukaryotes, so again, the operon system, this is only in bacteria, because bacteria need to put safe space and put the genes that are going to be working together all together with one promoter region. For eukaryotic cells, there is a luxury of having a promoter region for every single gene, okay? And it's way more complicated, as you can see here. So we have the promoter, we know that. We have the RNA polymerase. But then what is this, right? So it turns out that in order for a gene to get expressed, the RNA polymerase does not attach to the promoter by itself. It needs to be recruited over to the promoter site with things called transcription factors. Okay, transcription factors. These are other proteins that are made by the cell that are specifically for making a little bit of a a grouping, right? All these transcription factors come in one at a time. They bind to the promoter, and then there are other elements called an enhancer region. Don't worry too much about this. I want you to know the word transcription factors and how these guys are super necessary for RNA polymerase to actually bind to the promoter and to be stable enough to actually transcribe the gene that is in question. So for eukaryotes, it's a lot more complicated because we need all of these transcription factors. There's other elements up here. So the control of whether a gene is expressed or not is depends on not only RNA polymerase and the promoter, but it also depends on these transcription factors. And then these transcription factors, they can be activators, or they can also be repressors. They can turn on and off the gene. That is it. You survived.